Anyhow, we're going to start back up a little bit and start pickups. I want to go backwards a little bit, 2 or 3 slides. I didn't really, I don't think I've closed the loop on some things. Let me get the, I'm going to go back to here, and actually, frankly, I'm going to start by going all the way back to here, because I was thinking when I was eating a pear the other day that I might have misled you. I said that pears have these stone cells and apples don't. Well, apples do have sclerids or stone cells. They have them in the endocarp, which is kind of would be like the core, which is the part that most people don't eat. In fact, I would never eat the seeds if I were you, because about 50 will kill you. Okay, let's go back to, instead of starting right out in the new topic, I'm going to actually start here and close a few loops that I didn't close before. Now, we should finish this, what you have today, without a lot of trouble. All right, so one of the things that I didn't make, we were talking about lateral meristems, and the vascular cambium is one of them. And as we transition to secondary growth, that's what one of them, vascular cambium. Cork cambium is the other one I'm going to talk about this morning before we go back into the roots where we were. This, it's not written on the screen. It may be written in another slide or two, but there's a cambium that's inside the vascular bundle of a dicot, and it's called fascicular, because a fascicle is a bundle. And the cambium that's in the inside of the bundle is called the fascicular vascular cambium, and that vascular cambium that runs between consecutive successive bundles is called the interfascicular cambium, just like an interstate connects states. Okay, so that's just now the activity of that vascular cambium in time. Here it is on an open bundle, like you have in the open means it's open to further development. Closed is something like monocots. You don't get further development because there's no vascular cambium inside of the bundle or outside for that matter. Notice that there's none here. Now, I used to have some props or things I'd use when I do the show and tell when I would do stuff like this. And I was thinking, I wished I had my palm cross-section, palm tree, because when you look at a palm cross-section, maybe a foot in diameter, but there's not all this parenchyma tissue, not a real pith and cortex distinction, but basically it's parenchyma tissue. It looks like Spider-Man. That's kind of funny. The vascular bundle looks like some kind of Spider-Man image. But anyhow, vivid imagination. But anyway, back to the point. Something like Smilax, which I went out and cut one of these this morning, and look at the end of it, I tried to stain it. That's nothing but solid vascular bundles in cross section. You can't really see it, but in other words, there's not a lot of this. And then palm is the same way. There's just solid vascular bundles. I wish I could find a little tiki man that somebody carved from the vascular bundles of a palm and put on my desk. I never found out who gave it to me, but it's nothing but solid vascular bundles. Okay, now eventually, all right, the vascular cambium, fascicular and interfascicular, will form wood on the inside and phloem, secondary phloem on the outside. All right, now, and then we get to secondary growth, which is where we were kind of ending up, and I'm actually going to pick up again and make sure everybody understands a few things. I did send an extra slide this morning of something we'll talk about later. But secondary growth comes about by two means. the activity of the vascular cambium and the activity of the cork cambium. You know what I need to be doing? I need to be doing that annotate bit. Let's see. All right, now the secondary growth comes about by by the activity of this meristem, this lateral meristem, and this lateral meristem. The vascular cambium gives rise to secondary xylem on its inside and secondary phloem on its outside, to the outside. And obviously, the further out you go, the faster you have to divide to grow to keep up. That's why the phloem vascular ray gets dilated. It's kind of like a racetrack. You have to run further on the outside. It says here, the outer bark comes from the cork cambium and includes cork tissue. Now, whereas the inner bark is a secondary phloem. Now, I got these labeled a little weird because the cork, all right, there's a term that's on the handout I sent the other day called periderm. I don't think it's anywhere in this stuff, but the periderm is means around the derm, and the periderm consists of three tissues: the cork cambium and a tissue called cork or phellem, P-H-E-L-L-E-M, and another tissue called the phelloderm. Now, so in other words, the cork cambium and its two derivatives is the periderm. The cork cambium and its two derivatives constitutes an area that a region we could call the periderm. And peri means around and derm means the skin. So, but anyway, a synonym for cork is P-H-E-L-L-E-M, phellem. All right. Okay, I'm having that problem with the, all right, now, this is, again, one of my favorites, and it shows the inner bark is the phloem, and then the outer bark is what they're showing is the cork, but the phellogen is in here, and so is the phelloderm to the inside. So the phelloderm is inside of the cork cambium, and the phellem, which is outside. Now, phellem has some suberin, S-U-B-E-R-I-N, a compound that makes like a waterproofing. So that's why it's corky. And I don't have it in this presentation, but in the cork oak from the Mediterranean, they harvest this and use it to make real cork. It's a pretty interesting business. All right. So, all right, there's the product of the cork. Okay, now, and I talked about what this was talking about, the dilation of the ray here is because it's on the outside and having to undergo those anticlinal divisions faster than here to keep up. And the xylem parenchyma in the vascular ray is where the cracks form in wood. Now, all right, so we're getting closer to where we were. In fact, I think this is where we were, right? This is where we were ready to start. Are there any questions you can unmute and say, or any questions before we go forward with the new stuff, new business? Yes, go ahead. Steve's got a question. Yes. You are on this one or one other one? That one? Yeah. Okay. Well, it's secondary xylem. The primary xylem that was in the center has been crushed because of being obliterated, like you see here. The pith has been obliterated, and this is the primary xylem. Actually, no, it's the secondary xylem. The primary xylem that would have been right in here, along with the pith, gets obliterated by this secondary growth that's now solidified, is totally crushed to the center. So the pith is gone now, and so is the primary xylem that would have been there in the young days. Now, on this particular one, there's not a lot of distinction between, this diagram, there's not a lot of distinction between sapwood and heartwood. There is here. The heartwood is the part of the secondary xylem that has become occluded and filled up with compounds like lignin and other, lignin's a very, next to cellulose, is probably the most abundant organic compound on the face of the earth. This thing is full of cellulose, which is, again, the most abundant organic compound on the face of the earth. And the lignin is a hard derivative, hard form. Well, when you say lignified, it means hard. And the heartwood is significantly harder. Heartwood functions in conduction and in support, and the sapwood functions in conduction and support. Okay, does that answer your question, Steve? All right, any other questions like that or otherwise? Okay, now, all right, so now we're going into the root as an organ, root systems. The first slide will show that, and then anatomy, and then specialized roots. And today should be significantly easier than last Sunday in terms of comprehension, I think, because there'll be some repetition. But okay, we got the 4 major zones in a root that are easier. There's, of course, there's a root cap in a root that we don't find in the apical meristem of the stem or the shoot. Speaking of that sort of thing, I took my hat off earlier and looked, and then all the reflection was so bad, I thought I better put it back on. So that would be my apical meristem, so to speak. Now, okay, before we actually get into the regions of the root, this is sort of a difficult slide in a way, but where the slide is, I think I'm missing a slide. Yeah, I am missing a slide. Oh, there it is. Okay. All right. What, generally speaking, when you think of fibrous root system, you think of monocots like this grass. This foxtail over here has a fibrous root system. Now there are dicots that have a fibrous root system too, but this dandelion-like representative here is showing a taproot system. Now some taproots can reach all the way down to the water table in trees. Mesquite trees in the Midwest and West can have a taproot that can go 80 feet down to the water table, which is handy in the desert. So talk about adaptations. Now, Steve knows about why this darn machine is doing this sensitivity thing, but now, okay, this is we're going to talk about these, but first, this is a slide that it says an example of differentiated plants, not necessarily just roots, and it's true. We could be talking about the stem, but this brings up enlargement, number 1, elongation is number 2, nuclear division, number 3, cell division, number 4, and cell differentiation, number 5. Now, when a cell, when this cell is enlarging, it's enlarging in width, but it's also enlarging in length, it's elongating. Now, this enlargement continues until the surface area to volume ratio becomes less than favorable and the cell divides. Now, even in animals, cell division is a 2-part process. First, nuclear division, that is, a nucleus divides, and that's what's going on here when you have the new chromosomes going to the poles. And then, but in plants and in certain fungi, we have, actually all fungi, we have cell plates forming. Which is a precursor for a cell wall. The cell wall is different chemically in a plant from fungi, but anyhow, the cell plates forming here, laying down the primary wall that will separate these 2 cells. And the 2 daughter nuclei go to the poles and separate into the separate cell. And then at that point, the cell division is complete. Now, so what we're really saying is mitosis, which is what's going on here, is strictly speaking a nuclear division. I'm talking cell biology here more than I am anything else. So mitosis is nuclear division, and it's followed by cytokinesis, C-Y-T-O-K-I-N-E-S-I-S, better known as cytoplasmic division. So cell division consists of mitosis and cytokinesis. That's what cell division really is. And it's a two-part process. Okay, that's a fairly difficult concept. Any questions? Now, this differentiation business is, they're trying to signify that this xylem has gone an irreversible differentiation. At this point, that cell is no longer totipotent. It has evolved to a point that it can't de-differentiate and do something else. It's grown up into what it's going to be and can't change. Can't change jobs. And when you put on a secondary wall, that's part of it, because sclerenchyma is the same way. Once you sclerify, you've gone past the point of no return. Now, this is a tacky little drawing, but I like it. It's useful. It shows the root cap protecting the apical meristem underneath, and we're going to look at that again a little bit, and then this we have the root hairs in the differentiation region and a lateral root coming out. So at this point, there's some elongation. There's apical meristem, elongation, differentiation going on here, and at this point, you have the protoderm has become epidermis. The ground tissue has become the parenchyma and sometimes collenchyma that's in the cortical region. And then the innermost part of the cortex is called the endodermis, and that's what this is. The stele, a vascular skeleton or cylinder in a root, is identified first of all by the pericycle. So everything from the pericycle inward is a product of the procambium. Everything from the pericycle inward is a product of the procambium. Now, as normal in roots of dicots and less obvious in the monocots, which is what I sent you this week. And this morning, though, I sent you, actually this morning I sent you this, which you don't have as a slide. Okay, and that's the heart, that's the center of this, the stele. But okay, so we got xylem obvious and phloem not so obvious in this diagram, but in this diagram, it's a little more obvious. All right, so what you've got here is a cross section, and this is the slide I put in, and I told you I put it in last week, but didn't get to it, but I realized I hadn't really sent it to you, and I've got it here to try to illustrate, well, several things. One is, again, this is the stele. Everything inside of the endodermis is the stele. Now, it's Casparian strips. It's kind of a subtle concept here, but that's what you see here in this red out here in the endodermis. Now, notice as we go around the endodermis that the red or Casparian strips, which are full of suberin, by the way, the waterproofing I talked about on the cork a minute ago, suberin, that waterproofing in the endodermis is thicker on at the opposite I'm going to have to get some water, opposite the phloem, and thinner opposite the xylem. Thicker, thinner, thicker, as you go around. So here, look how thick the suberin is, and it's on the radial and the transverse walls. And what that is, the water that's coming in plant physiology, I got to get some water. Speaking of water, I got to drink some right now. Oh, excuse me. But when we talk plant physiology, which will be a few weeks, not next week. In fact, 2 weeks from now, we won't have it, and we'll talk about that. But next week, we will meet. All right. When water is translocating from the root hairs through the cortex and it gets into this point, you don't want to put water into the phloem. Why? Phloem does not conduct water. So that's why the waterproofing is heavy here. I'm actually talking plant physiology, which is hard to separate from morphology or anatomy. But here is where any water that's moved into here is going to diffuse over here and migrate into here, and then go up into the upper parts of the tree, or plant, in this case, buttercup. Okay, let's see. The phloem is never quite as obvious as the xylem because these cells have been stained. You could make your own slide like this with a buttercup root, and it wouldn't, this has been cut with a microtome and stained. Slide like that probably costs $2 or $3 or more now. They probably hardly only 1 or 2 places in the world even make stuff like this because people don't look at them anymore under the scope. These tons of these slides are sitting in mothballs in biology buildings around the world because nobody's looking at them anymore, but not many people. By the way, out here, you got starch grains in this parenchyma here. So that's what some of this stuff is, starch inclusions. All right, now, okay. All right, now this stele has only 3 arms instead of 4, like we just saw, but what it's showing is that the lateral roots are emerging from the pericycle. That is the primary function of the pericycle tissue, which is the outermost layer of the stele. It's also meristematic. That means it's capable of dividing, and that's exactly what it's doing. This slide got put in, as you can see. Actually, it didn't get put in until this morning, but I sent it to you Thursday or so, Wednesday or Thursday. And the reason I included it is because I for the first time, I shamefully recognized that I did not have a monocot root. And so that's what this is an attempt to do, is get a monocot root into the scheme of things. And the focus is, as I said in the email, I've got in the text of the slide in the PowerPoint, I've actually put the message I sent in the email. And the image came from that book I showed you this morning too. Actually, it didn't. It came from the big parent, from the big version of that. But in the little book that Donna's got, and I showed you botany book, it's in figure chapter 6, not chapter 9. But anyway, so the granddaddy of that little botany book is even worse in terms of what it's got in it. It's got enough for 2 botany courses. And that's enough for 1 botany course, general botany. But okay, now the thing that this slide and this slide have in common is they're both roots, but this is a monocot root. And what you've got in both of them is xylem, phloem, xylem, phloem. As you go around the circle, they are alternating bands. Okay, the phloem is not very obvious. Phloem, xylem, and it goes. But and in fact, let me go ahead and get rid of this one down here. This central, this weak cross section at D, as I said in the email. That's apparently a giant metaxylem vessel element that you say, gee, how do you know that's just not an airspace? Well, I think the wall here is well defined enough to say that it's not. But anyway, this is a wheat, which is a grass cross section of the root. This is an orchid, which they misspelled the genus name. Should be Epidendrum. Or epidendrum. This might have been what they call an orthographical variant, but that's now. But all the 2 photomicrographs on this giant slide the A and B are the most important. B is an enlargement of this region, and the phloem. You know what? This thing is right in the way, but right up in here somewhere. I'm going to have to take this. I'm going to have to take this annotate off for a moment, if I can. Try to. Well, I want to stop it. I think I did, but I'm going to have to stop this. Well, what I'm trying to show you is something I can't see because this thing is getting in the way. But up in here is actually a seed plate. Can you see it? Looks like a shower drain. I don't think I can. You know, let me exit out it. Oh, you know what? All right, let's see. Yeah, I can. Oh, there it is. See it? There's looks like, although this is right next to the xylem, which makes me suspicious. But what you're looking at is very much what I would expect to see in the phloem tissue. But the phloem should be here and in here, and occasionally you see in these companion cells. See that? Those are companion cells, and have to be, or it could be a xylem parenchyma cell, but you get a hint of the cell plates once in a while. But that looks like the kind of thing that I expected to see. Now I did drag that thing. Let me put it back up here, and now I can go back into my thing. All right. So there's a lot of silence here, except for Steve. I hope the rest of you are awake. Anyhow, that's what's the scary part of this, doing this kind of procedure is. But
Toot toot.
what's that? Oh, Donna's saying toot toot. Yeah, last week Donna kept it on audio and kept me knowing that y'all were alive. All right, now, all right, I'm hitting the, oh, you know what? This is so tricky. Okay, there it goes. All right, now these root function, this thing's in the way. I totally got rid of that when I did that, though. Can you see this well enough? This little arrow? All right, let's try doing it, because what happens when that toolbar goes in, the whole title gets blocked on mine, not yours. Root modifications and their functions. I'll try to wiggle it a little more because it's not quite as obvious as that little wand. But anyway, all right, so now this is pretty much easy peasy. Actually, that's quite complicated when we get to mycorrhizae, but this specialized business is not too difficult. All right, one of the major functions of roots, besides the other functions we mentioned, which absorption of water and minerals is a biggie, is storage. And we've got several here, got carrots and radishes and turnips and beets, parsnips. I'm not sure what this one is, but this is a cassava or manihot, the stuff that tapioca comes from. And that might be, this is like taro, but they're all earth vegetables, or they're for storage of carbohydrates. Now, the mycorrhizae concept is, all right, I'm going to go off on an important tangent that I mentioned once before, but I want to make it real clear. It's an example of how the English language changes over time, and sometimes not for the better. The concept of symbiosis today is not the concept of 1970, which was the heyday of ecology, as far as I'm concerned. But symbiosis is defined as the living together of two entirely different kind of organisms. Whether the nature of that relationship can be explained by adjectives that precede the word symbiosis. What you're looking at here is mycorrhizal associations of roots of higher plants with fungi, and it's called mutualistic symbiosis. And it's described with two plus symbols, a plus for the fungus and a plus for the plant. The less well-known, the part that might surprise you is parasitism is a form of symbiosis. But the news media wouldn't tell you that. They're part of the problem. They've caused part of the confusion. In fact, if you Google symbiosis now, you don't get the correct and original interpretation, because they think cloud sourcing or whatever has come to make mutualism synonymous with symbiosis, and it ain't so, people. Wasn't supposed to be so. But how would you describe parasitism? Not a plus plus, it's a plus minus. And the host is the minus. And that's not illustrated here. And the third and the final type of symbiosis is called commensalism, COMMENALISIM. And a good example of that, and that's a +0. And that good example of that is Spanish moss out there on my electrical lines. It doesn't need to be on, it doesn't parasitize a tree. And the fact that it lives on electric lines tells you that, and is a nuisance at that. But now I've got it on my pecan trees, and it harms them, not from parasitizing, but it harms them from the weight, and most importantly, from shading the buds and the damage. So the damage of Spanish moss is not directly as a parasite, but indirectly. So, but it's not technically a parasite, it's a commensal. Now back to this. That's an important ecological concept that I just brought up. So, but when fungi form relationships with the roots of high plants, they can do it within the cells, endomycorrhizae, or on the surface of cells, ectomycorrhizae, and illustrated in the drawings and in the photomicrographs. I love this slide because it shows a root, lateral root or rootlet covered with mycorrhizal hyphae. These are hyphae. Hyphae, which is the plural of hypha, Hypha. And you have to think about what a tremendous increase in the absorptive capability of water and minerals any root that is covered with this stuff has. It exponentially increases the surface area to volume ratio ability of this root to take up nutrients. And as we're going to point out in a moment, any plant that has the connection with mycorrhizae does better. And angiosperms in particular do better, and so do gymnosperms, as the next slide will show. Anyhow, okay, now who does what for what? Well, the fungus provides water and minerals from the soil for the plant much more efficiently than it could do by itself. The plant provides organic nutrients such as glucose. When we get to plant physiology, the primary product is a C6H12O6 glucose molecule. And it's called the photosynthate. And that compound is, I've heard, it's hard to believe that up to a third of the photosynthetic products gets consumed by the fungus, which is bordering on parasitism there, but this is supposedly mutualism. And obvious, the next slide will show that the plants do better that way when they form these relationships. Now, another thing you might be surprised to learn is that the fungus, in some cases, will make antibiotics for the plant. Isn't that neat? It's like having your own pharmacy. All right, but anyway, now here's this slide, and this slide shows how much better the gymnosperms are when you form relationships with mycorrhizae. This is a 6-month-old Pinus radiata Monterey pine seedlings growing in prairie soil, and then without the mycorrhizae, they look stunted, same age, but look how much healthier they are with the mycorrhizae. And look at this little lodgepole pine seedling. The majority of the biomass of the organism is beneath the ground, and a heck of a lot of it is tied in with fungal mycorrhizae. Imagine this kind of thing. If you magnified what you're seeing here, it would be much more. There's a lot of fungal body here, people. In fact, when we think about a fungus anyway, the sporocarp, which is this part, and since this is a basidiomycete fungus, this is a basidiocarp. This is actually a Suillus is a relative of Boletus, the bolete, but the bottom line is that there's spores here, and this whole thing is about big as your hand, and it is the tip of the iceberg. The body of the fungus is beneath the ground. In fact, the largest organism on the face of the earth is not a whale, it's a fungus that grows several miles and is thousands of years old. A honey mushroom is one of the examples that I'm referring to. I'm having trouble losing my cursor from time to time. But anyway, okay, so these plants do better, these gymnosperms do better when they're forming mycorrhizal relationship with fungi. And actually, you know, when we get, I thought I had it here, but I think it must be under, I think it's actually under plant physiology. But I've got soybeans showing the same thing. And I thought I had it in this slide set, but it's in the slide set. I believe it's in plant physiology. But all right, now, so this is specialized stems and specialized roots all in the same slide. I wish it were separate, but it's not. But in a way, it's a good example because specialized stem is like an Irish potato. And these we'll actually, I do have another slide of it in about 2 or 3 over, a little more detail. But now let's distinguish between two things that get confused: a stem type known as a rhizome and a stem type known as a stolon. They both have adventitious roots. An adventitious root is a specialization within itself. You'd see them on poison ivy growing up the side of a tree. Adventitious roots are roots that arise from some other root besides the primary root, the parent primary root. In other words, like coming off of the stem. Adventive, so to speak. But a rhizome is a subterranean, horizontal, usually horizontal, not always, stem, but subterranean. Whereas a runner or stolon is like you find in a strawberry plant, which is illustrated here. It has a function of asexual reproduction. There's a plantlet coming off of this stolon, which is what stolons do. You get plantlets developing this way, and it's a quick way of making babies that are clones. Whereas the function of a rhizome can be, it can do that. It can form asexual reproduction. And I think I've got a slide or two that will show it. But rhizomes are often fleshy and provide storage. Tapin fibers are the major types that we talked about earlier. Now, two other confusing types that we'll see in, I think, a subsequent slide is onion and corns. An onion bulb is mostly made up of fleshy leaves, and the stem is down here in a very, next time you cross-section, or no, not cross-section, longitudinally section an onion in the kitchen when you're cooking, look and see that the stem is right down here at the base, and you can actually see the apical meristem. And what you've got is all these fleshy leaves coming up around it. But a corm, like you're in dahlias, or in, I think of the gladiolus, is a solid entity, a solid fleshy entity called a corm, C-O-R-M-B, or C-O-R-M. Scale-like leaves, that right there, you don't get leaves coming off of roots. All right, now this is, okay, here's more detail. Here's a corm and here's a bulb. The actual stem is right down here inside of these fleshy leaves. But this whole thing, if you section it, it looks the same as that outside, but it's a solid stem, corm. These are the eyes, which are the buds. Okay, now sweet potato, it doesn't have buds on the storage organ. In fact, the storage organ of the carbohydrate in the sweet potato is the root. The storage organ of the carbohydrate in the Irish potato is the stem, or one of the stems. And of course, the carrot is a storage organ down here. We're putting out lateral roots. Questions? None? I'll go on.
Say that again.
Yeah, so, okay. Talking about the sweet potato being a modified root, Irish potato being, or the white ones, or red ones for that matter, being modified stems. And the way you'd know is that the eyes Yeah, you don't find eyes on a sweet potato because the roots don't have buds. The stem that joins here does have buds coming off of it, but anyway. All right, so they have one thing in common. They both are storage organs. And sweet potatoes are in the morning glory family, whereas Irish potatoes are in the Solanaceae, which actually are two families that aren't that far apart taxonomically. Now, this slide is a transition slide, switching totally to stems in our discussion now. And you can see a better example down here of the corm and how it looks in cross section. The less common of the specialized stems that most of you probably have never seen and may not see anytime soon is this cladophyll. A cladophyll is a clade means stem and phyll means leaf, and this is a modified stem that looks like a leaf, and it adds greater photosynthetic area. The regular main stem is here, but this is a modified version of the stem. And here shows strawberry producing runners and tendrils on grapes for examples of modified stems. On this Irish, we have a rhizome with adventitious roots. And then potatoes here showing, actually I have a better example of that right here. Let's see. Now, yeah, this shows the regular stem on a potato, and the specialized stems down here are tubers. And we'll talk about when we get to plant physiology why it doesn't pay to grow potatoes in warm climates. It's by Peru was the original source. But all right, now this is from my yard. In fact, not the same species as the one I showed you earlier. Not the same as this thing here with these god awful thorns on it. I've got about five species in my yard. I'm not bragging, they're a pain in the butt. That between that and the fire ants, I have my hands full. I was hoping the cold winter would take care of the darn fire ants. 8 days of sub-zero sub-freezing didn't do it. But anyway, this shows the rhizome kind of stem modification on the smilax, and there's your regular green stem up above. Okay. But you can see that not only is this performing a storage function, it's also propagating new plants. This rhizome has a double function, storage and asexual reproduction. All right, now I got cattails on the left and the god awful water hyacinths on the right. They have two things in common. They both have rhizomes that functions in asexual reproduction. This one probably functions a little bit in storage, but these this is a offspring coming from this plant through by way of this rhizome. And then this rhizome is actually floating on the water and forms new plantlets. I remember when I grew up we'd go to Florida on vacations, and I would have to get out and sometimes pull the boat through tons of this stuff because it would just clog the motors. Water hyacinths, they're pretty flowers, but it's not a native plant, and it clogs the waterways, and they sprayed and screwed up the ecology. It was a big mess in Florida. But look at these lateral roots coming off. But and there's actually root caps on this too. It's hard to believe. Why would you need a root cap in the water? It's vestigial. All right, and here's one of my favorite little shots down here. Black needle rush on the coast is a rhizomatous plant. And you don't have to dig it up to find that out, because look at what's going on down here. See all these, 1, 2, there's probably 7 plants here, another 5, 6, 7, 8, 9, 12. You keep counting, so what's going on is asexual reproduction of rhizomes producing plantlets in this rush, member of the rush family. It looks kind of grass-like. In fact, that's why it's called graminoid, meaning grass-like. Juncus romerianus is a scientific name for black needle rush. I'm going ahead and tell you that if you're on the coast, don't ever stoop down when you're standing in it because you can perforate an eye or eardrum. It happens. Very dangerous. Now, we're moving now to woody tissues and stems. And this is the part that gets into plant identification. In fact, you could find stuff like terms in here, like in Harris and Harris, because they pertain to plant identification. This is a horse chestnut, which is a good example to show external woody anatomy, or actually morphology, I guess, since it's external. But a node is a place on a stem where leaves occur, and you got scars. And then an internode is a region between two successive nodes. Now, this is, what are these scars inside the leaf scar? Okay, they are vascular bundle scars, vascular bundle scars. And I actually have, a tree of heaven right here that actually has alternate leaves, and you can see the same thing on it. But, and it's got something else out here called lenticels too, which are lenticels, or are they labeled? They're labeled on, yeah, here they are, they're labeled here. But I've got cherry that shows lenticels on it. See these little scars? These are lenticels. This is about a 40-year-old specimen that I cut from Pennsylvania, cherry, one time, and it's a beautiful look at, but it's a good example of lenticels. Anyhow, okay, so I can't see it very well, show and tell, but you get the idea. Now, by the way, that tree of heaven has got a 2.5 foot projection on it. It's all 1 year's growth. On this horse chestnut, this is, all right, these are terminal buds, and the terminal bud scale scars are right here. And the terminal bud scale scars a year before are right there. So you can tell how old this branch is. This is 1 year's growth. And a good year, that's longer than it is on a bad year, drought year. But lateral bud is a miniature of the terminal bud. And in the terminal bud, you'd often, it's a mixed bud because you'd find flowers and leaves in it. But normally buds are thought of, most we think of buds, we think of them as vegetative or floral, but they can be mixed. But notice that in the axil, which, by the way, this is a good example of AXIL, axil, the angle between the main stem and the branch. And there's always a lateral branch occurs where a leaf was, and there's a leaf scar. I don't know why that's like, it looks like it is. I don't know what the big deal is here. I don't really think it looks quite like that, but it certainly doesn't look like that over here. But anyway, you can tell the leaf arrangement was opposite, alternate, or whorl. Which is it? Opposite, because the leaf was here and the leaf was here. So you can tell leaf arrangement on a leafless branch, people. And that's, you can do that pretty easily. But, all right. Now, and pretty soon we'll talk about leaves and leaflets. And you're going to find out that you won't find a bud in the axil of where a leaf was. Excuse me, a leaflet was or is. You're only going to find a bud where a leaf is associated with a leaf. So buds are associated with leaves, not leaflets. That's the point. Got to take more water here. Now, look, we're focusing on leaves at this point. One of my more favorite topics. What you're looking at is a leaf of a black cherry. And it's a complete leaf. To be a complete leaf, you need three things. You need a blade. The cactus doesn't have a complete leaf because it doesn't have a blade. Spine is what you've got. Cuts down on water loss. All right, the second thing to be a complete leaf you need is a leaf stalk or petiole. And the third thing to be a complete leaf is stipules, and they normally come in pairs. It won't be long on this plant before the stipules fall off. Some stipules are persistent and some are not, okay? Next year's axillary bud is not yet showing, but it's there. So it's pretty well microscopic. Now, this is the same deal on a compound leaf, all right? So on the compound leaf, you have a trifoliate compound leaf, and there's no buds up here. There's no bud here, because these are leaflets. The bud is going to be down here, the lateral bud. A lateral bud is the same as an axillary bud, and it's called axillary bud because it's in the leaf axil, which is the angle between the leaf petiole and the main axis. All right, the stipules normally come in pairs, but they don't always come in pairs, as we'll see on the next slide. Questions? All right, none, we'll move on. Oh, the sycamore is notorious for having a fused pair of stipules into one large, actually leaf-like to a certain degree structure. Stipules on some members of the legume family can take the form of spines. But anyway, we have a big old stipule here, and we got a stipule here, and it's fused together. All right. Let me, this is, I love this slide because it it reminds me of what we used to do in the old days. We would take a lilac leaf or some leaf, and sometimes we use pith sticks, and sometimes put it on the end of on the glass slide, and take a sharp razor blade and roll a pencil or some dissecting needle backwards and make their sections to look at under the microscope. We'd make our own slides. And if you were good enough with a fresh blade, you'd get something that looked a lot like this. Ideally, you'd get a cross section of a vein, and sometimes you get a lateral section of a vein, and you could see the xylem a lot better in it, because in a pattern, if you section this, some of them are going to be longitudinal sections, and some of them are going to be cross sections of the veins. But last week, we brought up the term that we're going to elaborate on now, a set of terms. We said spongy mesophyll and palisade mesophyll. Well, there's the palisade mesophyll and the spongy mesophyll that's just called leafy mesophyll down here. Spongy is a better term. Palisade is like a fence, and they are fence-like. And they're closer to the surface, so guess who's going to get the most sunlight? These chlorophyll molecules inside of here, as opposed to these. As I said last week, mesophyll is a kind of parenchyma. In fact, you'd be correct to call it chlorenchyma. Why? chlorenchyma is a kind of parenchyma that has chlorophyll in it, okay? These terms make sense if you let them. All right, now we got a cross-section of a dicot, cross-section of a monocot, cross-section of the gymnosperm. Both top ones are angiosperms. Now, if you think about this, you'd have to go back to where we started this morning, but in a leaf, the phloem is going to be on the lower surface, and the xylem is going to be on the upper surface for the most part. Now, that's if that's so, this corn thing is upside down because here's the phloem and here's the xylem. But in this dicot, you got xylem down here. Let's see if I got that. No, I'm sorry. I think I got it backwards. This is, let's see. Yeah, this is actually correct. Because in a vascular, let me back out of this. Let me stop the sharing. Okay. On the outside of these vascular bundles is the phloem. That's here. Secondary phloem. This is secondary xylem. Now, if you took this stem here and went out into a leaf, the xylem is going to be on top. And the phloem, yeah, the phloem is going to, let me see. I'm having to think 3D here. All right. Yeah, the xylem is going to be on the top and the phloem is going to be on the undersurface. Now, with that in mind, the xylem was internal to the phloem. And so when you go outside, all right. Oh, shoot. All right. We're running out of near time here. Let's see, xylem. That's right. The xylem was on the inside of the phloem, and so it's going to be on the upper side. Over here, though, that just doesn't seem to be true because the phloem is up here in the corn. In corn, this is a vascular bundle. In corn, is basically bundles are veins. Okay, and so if the xylem in a stem is internal to the phloem, it's going to be on the upper side of the leaf. Because imagine a vein coming out of a stem and going at a right angle, or not a right angle, but a 45-degree angle. The xylem is going to be on the adaxial or upper surface, and the phloem is going to be on the abaxial or lower surface. That is not true the way this is oriented in this monocot. I didn't notice that before. But anyway, this is the vascular bundle in a monocot showing the phloem, and this is the vessels in the xylem of that bundle. And this is I believe that's an airspace right here with an occlusion in it. But and these are fibers that are associated with the bundle fibers. But over here in this dicot, we've got phloem fibers, we got secondary phloem, and we got the xylem up here. And then we've got these stomatal openings, which are in the epidermis. That's where the gaseous exchange takes place. In the case of the gymnosperm down here, these gymnosperms are xerophytes because they can survive drought a lot. And that's the substomatal chambers and the stomates themselves are recessed, whereas up here they're on the surface. And there's an epidermis has a cutin on it, a waxy protection from drying out on the dicot, on the monocot, and on the gymnosperm, you really have a heavy investment of protection. Now, another thing is gymnosperms do to cut down on water loss is put their veins in the center of the leaf instead of, this is a central tube, but the xylem where the water is lost into these chambers is closer to the surface. Whereas not only is the xylem in a conifer like this pine very much internal, there's an endodermis we don't find an endodermis in other leaves except for we do here. Now, how many veins are in this pine? 2. Some of them have 1. So taxonomically, the number of veins can help you identify pines if you make a section of it. But these are special kinds of mesophyll that have to do with preventing water loss in this pine. And this is a resin canal that we find in pines. We don't find in typical gymnosperms. But anyway, so we've got cross sections of 3 kind of needles. Now I'm gonna try to, we're running up an hour, but I'm gonna try to finish in the next 10 minutes. All right, we're on this, we don't have anything heavy duty from here on. Spongy mesophyll down here, palisade mesophyll here. It doesn't show up too well, but that's the same thing over here. Palisade spongy. Another, again, that's chlororenchyma, a special kind of parenchyma. This is a scanning electron micrograph. That's what SME of the stomata on the leaf surface. Most most of the stomates on dicots are on the abaxial lower surface. whereas dicots tend to have them often on both surfaces, but that's not, but generally speaking, putting the stomates on the lower surface is a way to cut down on water loss. And these little sausage-like cells become turgid or flaccid, full of water or empty of water, and that has to do with their opening ability. Some desert plants open at night, and that's a special kind of adaptation to desert life. We'll talk about that in plant physiology. All right, I like, let's see, I thought I had a picture of a lateral vein, a lateral view of a vein, but they're all cross sections. But anyway, these are just more of the same details. And these little guard cells are opening and letting, not only is water coming out, but carbon dioxide is coming in. Some of the carbon dioxide is manufactured into plant from respiration, but some of the carbon dioxide comes from the atmosphere. So the stoma not only lets water out, which is the way water moves up to the top of tall trees. Okay, this is a lateral view of a vein, not a good one, but anyway, cuticles, protection from water loss. Any questions? These are pretty busy diagrams, but they actually show these dermal, ground, and vascular meristems concepts that we talked of back when we talked about stems. All right. My Yankee friends think an evergreen is a Christmas tree. Well, the further south you go toward the tropics, the more you get evergreen angiosperms, not just conifers. And this is a good way. Once you find like Dahoon holly, which is barely in Georgia, but mostly more in Florida, it's more coastal in Georgia. But and the Fakahatchee of Naples, Florida, I'll be in a couple of weeks where I'll see John and some others. You'll have a lot of this. And in this stage of the growth, you can see that the new growth and the old growth is here at the same time. And it's very easy to understand that this is an evergreen plant. The leaves are on the tree. The average life of a leaf in the tropics is 13 months, they tell me. Leaf arrangement, this is not, if you do plant identification, you know what I'm talking about. Leaves are opposite, alternate, or whorled, and this shows opposite and alternate in simple leaves and in compound leaves. Node is a place on a stem where leaves occur. Internode is the region between two successive nodes. All right, pinnately and compound leaves. All right, this is a pecan showing what you normally have here. You have, there's no buds here. Why? These are leaflets. The bud's gonna be down here. Whoops. Once I lose it's hard to keep control of this pointer. Having trouble now. What happened to it? Okay, there it is. All right, the bud is here, and this is called a rachis. Again, CH is K, rachis, which is the continuation of the actually it's the would-be, let's just say this was connected all the way around by marginal tissue. And if we did, this would be a simple leaf, not a compound leaf. And look, and this is a juvenile pecan showing that fundamentally it is simple leaves. So as this thing gets older, it looks like these others, the compound leaf. But ash trees do the same thing. And by the way, you got 3 leaflets here on this one. So this shows that the simple leaf is the primary leaf and the compound leaf is a specialization of that. All right, Tacoma stands has another name, I think, but yellow elder is a tropical shrub that's in the Catawba family, has simple and compound on the same plant. These 2 slides are just full of terms, but sometimes spatulate is spelled with an H. But the point is, you would never sit down and try to learn all these terms. What you would do is when you key plants out, you would look them up. Now I know what they are because I've looked them up 1,000,000 times and I refuse to memorize them. So that's what the price I pay, but I've embedded them in my mind by doing that. And so there's just tons of terms to associate with leaves. Even when you get to when you get to this business here, mucronate, mucro, but if you look them up enough times, you'll learn what they are. So don't sit there and try to memorize them now, because it's a waste of time to my way of thinking. All right, what's the bad part about these drawings is nobody put buds down here to show you that the bud would be here, 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 and here. Notice this is something you'd find on a horse chestnut. Notice there's no rachis here. There's a rachis, there's a rachis, and there's no rachis in a simple leaf. But if you connected all these little leaflets together, you'd have a simple leaf. All right. Specialized leaves are like in the pitcher plant and this little sundew, they both are insectivorous. What they do is supplement their nitrogen, and therefore they can live in poor nitrogen soils, because chitin is loaded with nitrogen in insects. All right, and this is a Victoria Amazonica, used to be called Victoria regia, and you can bet your booties they put her down there with a crane, because she didn't climb up there, I guarantee you. That they actually got a normal water lily beside it. The flowers on the Amazonia aren't that much bigger than what she's got in her hand. I'm not sure that what she's got in her hand doesn't actually belong to the big one, but it probably belongs to the Nymphia odorata, which is there. But all right, the function of leaves can function in asexual reproduction, as you see going here. I've got some 50-year-old African violets in my living room that aren't violets, but the way you propagate them is take a leaf and stick it in the ground and it'll grow. But these are viviparous. That means living young plantlets coming from the leaf margin. Got roots on them, and they'll fall off of the margin. This is in the orpine family, the stonecrop family. But so asexual reproduction is a function of this kind of situation. So it's a way of cloning the plant real quick. All right, and that ends, they didn't do too bad, an hour and 8 minutes or so, but next time, and I'm gonna be sending you the slides for this, we're gonna focus on a Roman numeral 5C2. That's what this says. And when I say 5C2, I'm actually referring back to that one-page outline, or the slides 4 or 5 that are the summary slides. And in the way of homework, I'm not going to give you a matching, but the concepts that are going to be difficult will be sporophyll, sporophyll, which is seen here in a definition of a flower. And so that'll be a concept that we'll deal with. We'll deal with the pistil carpal distinction, and that's a difficult concept. And if you try to Google a definition of a flower on Google, you're not going to get what I've got here. You're going to get some simple dimple terminology for the layman, for the person that's not into this detail. But if we're going to try to elaborate next time on this concept of flower being a modified determined. That means that it ends at that point. Shoot, that's a stem bearing whorls of modified leaves called sporophylls, with very short internodes, with or without outer modified leaves. In other words, a flower doesn't have to have sepals and petals. We'll point that out eventually. All right, so we are done. I did I give myself an A-, except for my debacle with the leaf and the stem, and I tend to be a little dyslexic sometimes. All right, let me stop the screen sharing. All right, I hope we still got everybody. Yeah, it looks like it. And I hope you got something out of that. Let's see if there's any questions down in the chat thing. No, that's fine. Okay, here, bye-bye.
Thanks, Bobby.
Bye.
Bobby.